So now I want to talk about it's like the individual enzyme classes. I want to give you, it's like always a short introduction into the enzyme class, and I want to show you some of the reactions which can be catalyzed by these enzymes. So let's start with oxidoreductases, enzyme class number 1. So as already the name says it, they catalyze oxidation and reduction reactions. I've mentioned it already that 25% of all known enzymes belong to the class of oxidoreductases. Most of these enzymes, they require a cofactor for activity. So this cofactor can be an organic redox coenzyme, or it also can be transition metals used, or both of them, for activating the enzymes. Unfortunately, we also have to consider the fact that we need a cofactor regeneration systems to recycle the cofactors. Because, for instance, in the case of nicotinamide cofactors, these are very expensive. And we have to recycle like this expensive cofactor in order to perform a suitable catalysis with it. Therefore, some of these oxidoreductases are used as wholesale systems, because it's like we do not need the cofactor regeneration system for these applications. And the most useful preparative applications of oxidoreductases comprise dehydrogenases and reductases. The second group are the transferases. As already mentioned, transferases catalyze the transfer of molecular groups from one molecule to another. So these molecular groups include, for instance, methyl groups, but also nowadays alkyl groups with methyl transferases can be transferred onto target molecules. Carbonyl groups, acyclic groups, glycosyl residues, nitrogen, phosphor, and sulfur containing groups can also be transferred by using transferases. So from our perspective, these transferases are versatile catalysts for organic synthetic transformations. From the class of transferases, the most important enzymes are the transaminases. And especially in the last 5 years, transaminases have attracted widespread attention for the synthesis of amino acids and chiral amines. These transaminases catalyze the redox neutral amino transfer reaction between an amine donor and a carbonyl acceptor. And therefore they require PLP, pyridoxal 5-phosphate, as cofactor. And this PLP acts as the molecular shuttle for the transfer of the amine moiety from the amine donor onto the amine acceptor in order to produce the chiral amine. The biggest class of enzymes, as already mentioned, are the hydrolases. And these enzymes catalyze the hydrolytic cleavage of CO, CN, and CC, and some other bonds. So these are the favorite class of enzymes for organic chemists. Well, there is also a certain reason for it, because it's like those hydrolases, they do not need a cofactor. So in contrast to the oxidoreductases, where we always have to consider the cofactor regeneration, in the case of hydrolases, we do not have to deal with a cofactor and the regeneration of it. And furthermore, a large number of readily available enzymes of these hydrolases are available nowadays. Because it's like the hydrolases were the first enzymes people have worked on 30 years ago. And therefore it's like much has been done on the identification, discovery, and study of hydrolases. Hydrolases can also catalyze the reversible reaction. So we can use these hydrolases in organic solvents to produce amide or esters. And these hydrolases have a relatively high stability under non-natural conditions. That means that we can use them in organic solvents. And furthermore, those hydrolases often show a wide substrate range. So it's like these hydrolases have everything we have dreamed of when we were thinking of a biocatalyst. The fourth class are the LYASES. I already mentioned there's a certain interest in increasing the toolbox of available LYASES, because LYASES catalyze the cleavage and the formation of CC, CO, and CN or other bonds by other means than hydrolysis or oxidation. So it's like the CC bond formation is one of the pivotal reactions in organic synthesis. These LYASES are highly regio- and enantiospecific. So excellent yields and enantiomeric excesses are possible or reachable by using these LYASEs. These LYASEs are applied in redox neutral reactions So it's like sometimes it's like no cofactor, so there is no cofactor regeneration needed, in contrast to the oxidoreductases where also cofactors are applied. Many LYASES contain prosthetic groups, and because it's like also the need is like these prosthetic groups as cofactors in order to achieve the activity or reactivity which is needed for the catalysis. So the next class are the isomerases. As already said, it's like these isomerases are applied in the interconversion of isomers. And so we have geometric or structural changes within a molecule. And these isomerases comprise a whole set of enzymes called epimerases, racemases, cis-trans isomerases or tautomerases. So it's like many enzymes are part of this isomerase toolbox. The reaction mechanism depends on whether the substrate, in most cases in nature there are amino acids or sugars known as substrates, whether it's like the substrate is activated or not. And usually it's like we have basic amino acid residues which are involved in the catalysis by cleaving certain bonds, CH bonds.